So, quite good presentations. I'm just observing. Dr. Ali Asgar, what he has said that it's difficult to sell concepts unless you have become a brand. The point is that, yes, it is really difficult to sell concept unless we have some brand with us. The main issue is that we are talking about sustainability in poultry production. Sustainability everywhere, wherever you go, in any conference, any seminar, you will find that sustainable poultry production is the main thing. Now, how this sustainability can be achieved? This is a big question. The genetic makeup of the modern birds is so high, it's so sophisticated nowadays, that if there is a bit of problem in anything, be it in the feeding management or in the or in, you can say that simple management system, there will be some problem and we will lose this sustainability we are talking about everywhere. Now, to keep this poultry production sustainable, be it in broiler chickens or it in case of laying hens, we need to think about something that how to maintain the feed intake intact. Because, this nutritional psychiatry is one of the most important points that everybody should keep in mind. Nutritional psychiatry, what does it mean actually? What is nutritional psychiatry? If I ask you a question that, well, you need to take a flight, say now it's just 11:45 and you need to take a flight at say 12:45 and you are still stuck up in a traffic jam. If I offer you something very good food, will you be in a position to enjoy it? Or will you be even think of going for it? I'm sure that you will not, unless you are sure that you're going to miss the flight for sure. So this is called the nutritional psychiatry. This nutritional psychiatry is something which actually is being controlled by the brain, the small intestine, as well as the bacterial population which are present within the small intestine. So these three things actually play consecutively to control the feeding behavior of everybody, be it a human being or animals. So these three must play in consequence so that this nutritional psychiatry can be maintained properly. If the brain remains happy, the bacteria which are present in the small intestine, they also remain happy. And if the bacteria remain happy within the small intestine, they keep on sending some signals to the brain, and the brain actually starts feeling that, well, I'm happy, so I can now eat. And if this eating behavior, the feeding behavior continues, in that case, that should translate into some sort of sustainability, because unless the bird doesn't consume, you will not be in a position to maintain the body weight and performance. So this is a very important thing. Now, this communication between the small intestine, the bacteria, as well as the brain, that actually continues by means of something which we call that dialogue process. A dialogue process always goes on between the brain of the host animal, the bacteria, as well as the physical structure of the small intestine. Generally, what we consider that the bacteria, they enjoy some sort of commensal relationship with the host. But actually, it is not commensalism, but we should say that this is a mutualistic relationship that the bacteria enjoy with the host animal. Why it is mutualistic? Because the host animal, it gives the bacteria some sort of hospitable niche as well as some undigested food. And once the bacteria, they grow inside the animal, what they do? They synthesize some sort of neuroactive peptide compounds, neuroactive compounds. These neuroactive compounds, they impact the central nervous system, especially the enteric nervous system, and this enteric nervous system, and finally, they modulate the small intestine structurally as well as functionally. So that means these bacteria which are present within the gut, they enjoy the food from the animal, and at the same time, they modify the small intestine structurally and functionally so that the host animal thinks about continuing eating or stop eating. So this is the main point. That means it is difficult to say that the bacteria are always enjoying some sort of commensal relationship. Rather, it is a mutualistic or mutualism which actually is going on. Now this dialogue process, what I'm talking about, between the host animal, between the bacteria as well as the small intestine, that actually is mediated by a number of neurotransmitter substances. The neurotransmitter substances, they are of different kinds. For example, they may be excitatory, they may be inhibitory. There are some neuromodulator substances, and there are some neurohormones. So these substances are being secreted by the bacteria which are present in the small intestine, and finally, they secrete some sort of secondary mediators. The secondary mediators are actually some sex steroids. They may be adrenocorticotropic hormone, maybe corticosterone, ghrelin, peptide YY, and so many things are there. There is endless counts which are being secreted by the body itself, which actually control the final feeding behavior of the host animal. For example, let us consume a situation. The situation is something like that the bird is being fed with something which is not really digested. That means the feed is being stored in the small intestine for a long period of time. Under that situation, the bird will need that, okay, if the feed is stored for a longer period of time, it needs some sort of enzymatic action, or you can say that some overexpression of the enzymatic action actually, which will help the feed, undigested feed, to be digested easily. Under that situation, the neuromediator substance peptide YY will be secreted by the bird. This peptide YY is a substance which will put some sort of ileal brake. That braking mechanism will work. The braking mechanism will not allow the food to pass through the hind gut. So it will remain for a longer period of time within the gut. It will be exposed to the enzymes for a longer period. And as a result of this, the feed will be digested. So this is how this dialogue process goes on. The host controls the digestion process, and this digestion process in turn is being controlled by the bacteria which are present there. Now, how these neurotransmitter substances work? These neurotransmitter substances work by sending some sort of signals which actually pass through the neural junctions. The neural junctions means there is one neural junction where the neurotransmitter substances come, and then it moves on to the second part, or that means the second neuron. Now this process, this area is called the postsynaptic neuron. This is the presynaptic neuron. Now here from the action potential actually moves from one neuron to the next neuron. And actually it depends on what type of the neurotransmitter is in operation, which actually will control that whether this action potential will move from this neuron to that neuron or it will not. Now there are, as I have told, that there are types of neurotransmitter substances, for example, excitatory or inhibitory. If it is as excitatory neurotransmitter, for example, epinephrine or norepinephrine, then this action potential will move through a gate. The gate will become opened, and as a result of this, the action potential will move from one neuron to the next one. But if it is an inhibitory neurotransmitter substance, for example, gamma-aminobutyric acid or serotonin, the gate will become closed. And the gate becomes closed, in that case, the action potential will not be transferred from the one neuron to the next neuron. That means that there will be some sort of inhibitory or inhibition effect. Now, it is interesting to note that these neurotransmitter substances are not always secreted by the host animal itself, but the neurotransmitter substances are secreted by the bacteria which are present in the small intestine. For example, this dopamine, norepinephrine, serotonin, acetylcholine, all these neurotransmitter substances are secreted by a number of bacteria which are there itself in the gut. And out of this bacteria, there are a number of bacteria, for example, lactobacillus or bifidobacterium or lactococcus, which are being considered as the beneficial or friendly bacteria for the animal. Now, this brain microbiome dialogue, how does it work? Now, there are a number of models. In case of human beings, the model says that the gut microbes produce some neurotransmitters which travel to the brain via the vagus nerve and then sends a signal to the brain, and the brain accordingly works. The second mechanism which says that these, depending upon the situation, for example, if there is an impending infection or impending threat of some attack on the small intestinal wall, in that case the microbes, they will secrete some neurotransmitter which will stimulate the brain to secrete more amount of the cytokines. Cytokines means which will fight against infection by means of, they're providing immunity. And the third mechanism says that they will produce some metabolites which will travel through the blood or will stimulate the vagus nerve to send important messages to the brain, and the brain will then act accordingly to control the feeding behavior and to sustain the growth or not. Now, if this is the concept that if these neurotransmitter substances can be translated into a product or into something which can be supplemented externally into the bird, in that case probably this question of sustainability can be solved to a great extent. Now, if we go for searching these neurotransmitter substances, there we should search for something which is the right candidate. Now, what may be the right candidate? The right candidate, they should have these properties. For example, they should be inhibitory in nature. They should not allow the neurotransmission to pass on from the one action, from the one nerve ending to the next one. They should have some blunting effect on the action potentials. And finally, they should have some calming effect on the brain so that it can sequester the inflammatory responses that is present in the gastrointestinal tract mucosa. The main objective of preparing or coming up with such a product is just to conserve the nutrients which will ultimately be used for growth promotion. Now, one of the candidates is gamma-aminobutyric acid. Why this is so? This gamma-aminobutyric acid is basically a neuroinhibitory substance which is produced by some brain cells, which is produced locally in the small intestine, as well as it is produced locally by a number of bacteria which are present there. Now, this is the main mechanism of action of this gamma-aminobutyric acid is that it inhibits the acetylcholinesterase function. This acetylcholinesterase function is such a function which actually is inhibitory for the growth of animals, because it in turn stimulates the renin-angiotensin system. And if the renin-angiotensin system is stimulated, actually this down-regulates the insulin-like growth factor functions. What this gamma-aminobutyric acid does, it actually inhibits the acetylcholinesterase system, and as a result of this, the insulin-like growth factor function is upregulated. And once this insulin-like growth factor action is upregulated, we can expect there will be protein accretion, so there will be increased muscle mass, performance will be more, there will be more there will be more burning of the body fat. So burning of body fat will provide more energy for the amino acids to be incorporated into the muscle mass. And finally, there should be more performance enhancement. So this is how this gamma-aminobutyric acid may be considered as one of the candidates which can be explored to impart sustainability in future poultry production. Now, these inhibitory properties of gamma-aminobutyric acid is actually quite interesting because it can stimulate the feed intake. As I was saying that this neuroendocrine behavior is very much important in sustaining the poultry production, because one needs to maintain the feed intake properly under different condition. This is very interesting because you can see here that in number of breeds, this black bird is for the cob and the grey bird is for the Ross breed. So in both the breeds, this gamma-aminobutyric acid, it stimulates the upregulated the functions of two orexigenic genes. One is the agato-related protein gene and another one is the neuropeptide Y. So both these genes are upregulated under the influence of this gamma-aminobutyric acid. And this happens even in presence of heat stress. So this is possible that with this gamma-aminobutyric acid, the feed intake can be stimulated even in absence of or in presence of stress. Now, if we consider some detail of the effect of the gamma-aminobutyric acid, which is one of the inhibitory substances, so this actually a number of research has been conducted, and more than 80% of the research says or shows that under the stressful conditions, especially when the chickens are exposed to heat stress, application of gamma-aminobutyric acid can stimulate feed intake, can promote growth, as well as can improve performance, the feed conversion ratio. Now, this is another example that how these inhibited neurotransmitter substances can check infection or you can say that can stimulate immunity. This is important because I'm just picking up here this gamma aminobutyric acid as an example because the results are available with this molecule only. Now, if I consider this one, there is one direct effect. The direct effect means it stimulates the synthesis and secretion of the secretory immunoglobulin in the small intestinal mucosa. So, if any product is there which can stimulate the synthesis of the secretory immunoglobulin in the small intestinal mucosa, that is going to benefit the bird's health because it is providing some sort of local immunity. And at the same time, it is going to prevent the loss of that nutrients by means of inhibiting the peristaltic movements in the hind gut. So if the peristaltic movement in the hind gut is restricted, in that case the bird will lose less amount of water, less amount of fecal materials because of these constrictory properties. As a result, there will be conservation of the nutrients, and so there will be a couple of benefits with the application of this inhibitory neurotransmitter substance. Now, there is one confusion that whether increasing the dose of an neuroinhibitory transmitter substance is ultimately beneficial or not. And if what will happen that if it continues for a long period of time, to be honest, the data is not really available to that extent. But so far, within a short limit of dose effect and a short limit of time effect, the available data actually shows that there should not be any negative impact of applying the neuroinhibitory substances in the feeding regimen or in the drinking regimen of chickens. For example, if incremental dose regimen is applied, in that case, body weight gain, feed conversion ratio, as well as immune properties which was measured in terms of immunoglobulin levels in blood, both are improved, all are improved. Similarly, if keeping the dose level same, if the duration is increased, in that case also, positive effects on feed intake, feed conversion ratio, as well as immune properties like immunoglobulin level, CD8 cell activities, the activity of the spleen as well as the bursa, everything was improved. So, depending on the available literature, available data, it can be said that within a short period, within a short dosage regimen, and within a short period of application, it is possible that the neuroinhibitory substances can improve the performance even if it continues for a longer period of time. Probably this is going to be the future of the products or future of the concept. Now it is possible that there are neuroinhibitory substance producing bacterial cells which are present in the small intestine. So by means of genetic engineering, it is very much possible that those particular segments can be cut, those segments can be put in a carrier bacteria, and finally this carrier bacteria can be converted into a big product which can be supplemented to the birds, and finally this may help in sustaining the performance in future. So if I try to sum up, so this may be something like that the candidate molecule which is going to be developed in future, so they should have something, they may be applied from the outside or they may be developed inside, whatever it may be, they should have a complementary action. And finally, they should have certain properties. For example, it should improve the local and the systemic immunity, it should sequester the pro-inflammatory changes in the gut mucosa, it should upregulate the orexigenic genes so that the feed intake continues, and finally, it should have some positive effect on the performance in terms of body weight and feed conversion ratio. That's it. So if there is any question, you can ask me.
Hello. Thank you, Dr. Sudipto.
Thank you very much.
I like that communication between gut, microbes, and the brain.
Main thing is that whether I could communicate well with you.
Thank you so much. Yeah. So any questions for Dr. Sudipto? Okay, fine. So he is around if anybody wants to.
Yeah, you can reach me anyway.
Thank you so much. Thank you so much for the presentation. So may I now invite Meghal Singh for her presentation on farm to fork digitalized approach. Meghal. Sorry?
Thank you, sir. There is a very brief announcement. We are very happy to have Dr. Gaura Dubey ma'am here, and she is the chairman of Phoenix Group, Jabalpur. We welcome you, ma'am. And I request Dr. Lakshmi Chauhan from Department of Poultry Science to please welcome Gaura ma'am.